Good afternoon, everyone, and welcome to this Genome webinar. I'm Ben Butkus, Editorial Director at Genome Web, and I'll be your moderator today. The title of today's webinar is A Clinically Validated and Easy to Implement HRD Scoring Solution Using a Targeted Panel and Shallow Whole Genome Sequencing. It is sponsored by SeqOne Genomics, which provides an integrated platform for germline and somatic genomic analysis in clinical environments. The topic of today's webinar is homologous recombination deficient, or HRD, tumors. Pascal Pujol, head of oncogenetics at University Hospital of Montpellier, will first discuss the latest thinking on HRD detection. Following this, Michael Blum, Director of Research and Development at SeqOne, will present the company's newly launched HRD test. Attendees may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are a series of widgets to enhance your webinar experience. With that, I'll turn it over to Pascal Pujol of the University Hospital of Montpellier. Please go ahead, Pascal.
Thank you very much, Ben. It is my pleasure to talk today about HRD. I'm a clinical geneticist working at the University Hospital of Montpellier. We have to define what we are talking about when we're talking about HRD. And this is my disclosure of interest. The definition of HRD is the inability to repair properly double-strand DNA breaks via the homologous recombination pathway. We have to remind that DNA lesions is a common feature of cell life. It's happened as much as 50 times a day for a single cell, depending upon cell cycle or the type of tissue. And these type of events are even more frequent in cancer cells. DNA repair pathways is a very complex process, and I will not go through all this process. Let me reassure you for that. But just quickly remind that we do have repair pathway specialized in a single strand break. And we have to mention here an enzyme called PARP. We also have double strand breaks repair pathway. The well-known homologous recombination pathway involves several genes, as BRCA1 and BRCA2 gene, and several other genes. And in parallel, we have what is called a non-end joining pathway outside the HR pathway to repair double-strand lesions. And finally, also, I would like to mention another process for mismatch and excision repair nucleotide for a single mutation or deletion. Homologous recombination involves many genes and proteins, and they interact at different steps of the process. I will not go through this again, but just let me mention very important protein and well-known like BRCA1, BRCA2 gene, PALB2 gene, ATM, and the RAD50 family. Something very important in terms of therapeutic point of view is to remember that if you inhibit the repair of a single strand break, for instance with the PARP inhibitor, you will have at replication during the S phase, you will generate double strand breaks. In normal cells, HR pathway will be able to repair. But in cancer cells, where HR is deficient, like in BRCA mutated tumor, that will lead to genomic instability and finally to apoptosis. This is a very important mechanism called and concept called synthetic lethality, which is very crucial to understand the topic of today about HRD. Now let's switch to the second part of this talk. Is why should we have to measure HRD in clinic, and particularly in ovarian cancer? Let me just remark that HRD is not an ubiquitous mechanism. We do have HRD in at least 4 cancers so far. Breast, prostate, and pancreas cancer account from 5 to 15% of tumor with displaying HRD. And in ovarian cancer, it's up to 50%. The most common cause of HRD is the mutation of BRCA1 and BRCA2 genes. It could happen both at the germline level or sometimes only in the tumor. We call that somatic mutation. PARP inhibitors was first used in clinical study in ovarian cancer according to BRCA mutation. And the results and the reason why we are here today is because these fantastic results. We are talking about maintenance with olaparib, which is a PARP inhibitor, versus placebo in BRCA mutated ovarian cancer. And as you can see, olaparib dramatically increases not only PFS, but also overall survival. This is maybe the most striking results in solid oncology since a long time. In placebo group, the PFS was 13 months. In the olaparib treated group, the PFS was 56 months. So what we can say from this is that BRCA status is crucial, not only to test and to predict sensitivity to PARP inhibitor, but to treat ovarian cancer women. The step after, from a clinical point of view, was to see if there is any benefit beyond BRCA mutation. And this was done using the defect of the HR pathway. In all studies available, you can see that we have a benefit in women having tumor with a deficiency in HR beyond BRCA. I will not go through all the study, but just pick 3 of them. The first one is a Paola study comparing olaparib plus bevacizumab to see the benefit in ovarian cancer first-line maintenance compared to placebo. And as you can see, the result is also very significant. Beyond BRCA woman, the woman with HRD have also a great benefit from PARP inhibitors. But in non-BRCA mutated tumor and non-HRD, there was no benefit at all. It is of note that this woman was not selected on platinum response. The second phase 3 trial is a PRIMA study comparing niraparib versus placebo in ovarian cancer maintenance. Again, you can see a gradient in between BRCA mutated women and non-BRCA mutated women and non-HRD women. But we have to note that patients here were selected on the platinum response. And finally, this is the most recent result of the Athena mono trial comparing rucaparib to placebo in ovarian cancer maintenance. Again, we have the same kind of results with a very important benefit in mutated women, benefit in HRD women, and a weak benefit here in non-HRD women. But in this study, it is important to say that HRD was defined on the basis of LOH. So it could be that there is some missing HRD when you are looking at LOH. We will go back to this point later. Well, to summarize, I mean, in all studies available, we have benefit in HRD subpopulation. And so the consequence of that has been stated in the HRD European consensus conducted by Ignace Vergote and published last year in Annals Oncology. Biomarkers of HRD are effective to predict sensitivity to PARPi in the maintenance of ovarian cancer and should be carried out in all women with epithelial ovarian cancer at time of diagnosis. And now let's move to the last part of this talk. The question is, how should we measure HRD? We have two ways to do it. The first one is to look at the consequences in terms of genomic scars of HRD. And the second one is to look at the potential causes of HRD. Let's start by looking at the causes, as far as we can understand the causes, actually. I mean, we mentioned that BRCA1 mutation, both at germline and somatic level, is a very good way to predict PARPi sensitivity and HRD, by the way. Another way could be to look at other genes involved in HRD. Another way also to look at epigenetic lesion of this gene, such as methylation of promoter, especially in BRCA1 and RAD51C gene. So since that has been described. Okay, so this is a way to look at causes, and there is a lot of study about that. I will go back to this point. Another way is to look at the consequences, and the consequences of HRD is genomic scars. So why don't we look at the genomic scars? You know, a simple way to do it is looking at loss of heterozygosity, LOH, and it works. Another way is to look at genomic instability score. This has been done in many studies by looking with a solution that came from Myriad Genetic. It's called My Choice. And actually, it's add LOH plus what is called telomeric imbalance and large-scale rearrangement. So you sum these three alterations, and you've got a score reflecting instability of genome inside the cell. Finally, a very smart and elegant way to look at HRD is to look at functional consequences of HRD. I will quickly go back to that, but you can look at foci of RAD51, which is a very important protein, crucial to recruit the actor of HR repair. Another one is to look clinically at platinum sensitivity, which is a very good marker of HRD. And finally, to look by sequencing whole genome at this stability. Let's start by looking at HR gene. Is this a good way to define HRD? The answer is no. Because if you do that, you will miss a lot of HRD patients. HR mutation in large panel of gene just account for a small part of HRD, ranging from 3 to 6%. But more importantly, these mutations fail to predict olaparib sensitivity in many cases. This is a result of a study of my friend and colleague Eric Pujol that was published 1 week ago, actually 3 weeks ago, and that shows that the mutation of this gene was not sufficient to predict PARP inhibitor sensitivity. This is not very surprising because when you look at mutation on this gene, only a few ones have a high genomic instability score, particularly actually BRCA, of course, but RAD51C, PALB2. Just remember this actor because actually those ones are predicting of PARP sensitivity, but as you can see here, the other not. And this is confirmed by other study looking specifically at HR gene panels, and as you can see, it's a little bit disappointing. So my conclusion of this part is that if you look at scores, BRCA1 and BRCA2 are first in class. They are the best marker to predict PARPi sensitivity. And only a few genes among the panel could do that, namely RAD51, PALB2, BRIP1, but only 2 to 3% of mutation. So it's not sufficient. And it could be a little bit severe, but I will conclude by saying that HR gene panel could not be used in clinic to predict PARP inhibitors sensitivity beyond BRCA, of course. And this panel should be reserved to research use only. An elegant way to look at HRD is looking at the functional consequence, and especially looking at what is called RAD51-4C, will play a major role in HR repair pathway. And you can look at the absence of RAD51-4C at the DNA double-strand break. And this is a functional marker of HRD. This is in development. It's a very promising way to look at it, but unfortunately, again, we need a further confirmation, and it's for research use, in my opinion. A last way, I will not go to all HRD solutions, but just mention this, the work of this French woman, again, Anne Vincent Salomon, who is using algorithmic tools and IA applied to pathology to see HRD profiling. And they are very promising reason published recently with an AUC of 0.8 as compared to the myriad test. So it's encouraging, but again, we need some further validation for use of pre-screening of this solution. The last question before going to the very last part, which is how to measure genomic instability, is an important point. I mean, if you look at genomic instability, the solutions are not equal. Look at that. It's a comparison between LOH and the results of my choice. And it's not concordant, more than 0.84%. So that means that assays are not the same. And this is an important thing to remember because at this time, we do have a lot of solutions available. Some are coming from institution and academic milieu, and other from commercial and company. I mean, it's a great news, I mean, to have all these tests available. But we should be aware that we have to define criteria of quality of these different assays. This is the goal of a new ongoing European initiative called Joint Consensus on HRD testing that I have the privilege actually to conduct with a lot of friends from European countries. And the thing is, I mean, not only we have to actualize clinical guidelines, but we have to grade all these multiple companion diagnostic assays available. And also we have to take into account the resource of the country or of the structure, because some tests are very expensive. We aim to first evaluate and grade HRD companion assay for all solutions available. It includes what you call GIST, genomic instability, but also HR gene panels, pathology, etc. Signature. And for that, we have established pre-analytical, biological, and clinical criteria of validation. We will also take into account cost, access, reactivity, turnaround time, etc. For clinical update, we will use new data from long-term results of data on PFS and OS in phase 3 trial, and also consider new or modulation of FDA or EMA approval. This is very important that we already know that for validation criteria, there will be on the top of it one very important, which is the clinical validation. Even if we have very good rationale to use one test, very good correlation, we need that will be the best thing to have for a test, a clinical validation according to one of the phase 3 trial. There is emerging question also to address. Sometimes you do have a BRCA mutation, but you have HRP. I mean, no deficiency of HR pathway, and probably no sensitivity to PARP inhibitors. We have to think about that. Also, we've got another point. I mean, you have no HRD, you have a wild type BRCA status, but maybe there is still sensitivity to PARPi, and we need to go further solution to be able to detect this kind of tumor. Also, we are wondering about CCNE1 amplification and the clinical potential interest of it. So, Ben, let me conclude by this statement. I mean, a lot could be read in this excellent paper from Ignace Vergote. Biomarker of HRD are required at time of diagnosis of any epithelial ovarian cancer. They are needed because they are predicting sensitivity to PARP inhibitor. Biomarkers of HRD are not interchangeable and must have a clinical validation. That's why we are at this point to build new guidelines with a focus of HRD testing, and I think this is an excellent way to give the floor to Michael that will Blum that will present a new solution. Thank you very much.
Thanks a lot, Professor Pujol, to give me the occasion to introduce the HRD test we have developed at SeqOne Genomics. So it's a new test which is based on two assays, a targeted gene panel and shallow whole genome sequencing. So I do work in SeqOne Genomics, in which we develop a platform to analyze genomic data. So we assume that the wet lab and the sequencing is done in the lab, and then the SeqOne platform handles everything which goes from data transfer to the automatic generation of the report, typically a PDF report. And we provide compliant CE-IVD platform, which should be CE-IVD-R this year. So what's in the SeqOne HRD test? So as I mentioned before, to build an HRD test, we use two assays, gene panel, and we use a gene panel to call the BRCA1 and BRCA2 pathogenic mutation. So we have an automatic procedure for variant calling in BRCA1 and BRCA2, and then to determine the pathogenicity according to ACMG criteria. And the clinical validation, which I'm going to present, has been validating using the gene panel from Agilent. If the patient has BRCA1 or BRCA2 pathogenic mutation, it's HRD, and otherwise we have to go through shallow whole genome sequencing. And in the shallow whole genome sequencing, we use like 3 features. So the first one is we count the number of times there is a loss of parental copy. So in this slide, there is an example in which there is 1 loss of parental copy, so a ploidy that goes from 2 to 1 in one particular region. Then we also count the number of large-scale genomic alterations. So here in the slide, there is an example of 1 alteration where the ploidies goes from 3 to 2. And the higher the number of large-scale genomic alterations, the more likely it is that the tumor is considered as a HRD. And then we also look at another feature, which is the CCNE1 copy number variation. And the more amplification there are in the gene copy, the more likely it is that the individual is HRP or HRD negative. So it's the opposite direction compared to genomic instability. They work in opposite direction. And so we combine all this information, so BRCA1, BRCA2 mutation, plus all this genomic instability and CNV amplification in order to compute a single score, which is a probability for the tumor to be HRD. If the probability is larger than 50%, it's a HRD sample, and otherwise it's a HRP or HRD negative sample. So in practice, there are like 2 different approaches to run a SeqOne HRD. Either the lab can do a parallel testing approach, which is time efficient, in which 2 assays, the gene panel and the shallow whole genome sequencing, are performed at the same time. And there is a second approach, which is cost effective, in which the lab starts to run the gene panel to determine the BRCA1 and BRCA2 status. If it's mutated, it's a HRD sample, and if it's a wild-type BRCA sample, then the lab can perform the shallow whole genome sequencing. So now it's my pleasure to introduce the clinical result of the validation of SeqOne HRD, which have been presented last week in Barcelona during the ESMO GINAE conference. And it's a joint work with Romain Boideau from Dijon and the team from Arcagi that sponsors the Paola-1 cohort, including in particular Eric Pujol. So we use the Paola-1 cohort. So as said by Professor Pujol, it's a pivotal cohort to show the efficiency of olaparib for HRD patients. And we analyzed 368 patients from the Paola-1 cohort. First important result, we get a large, high agreement between SeqOne HRD and Myriad My Choice. So more than 90% of overall agreement between the 2 solutions, between the 2 sets. High agreement between the 2 solutions. Then second important result we get is we have 1% of a non-conclusive result with SeqOne HRD, whereas it's 7% of a non-conclusive result with a myriad matches. And one of the reasons that explains this difference of rate is that we can handle a low cellularity sample of 20% of tumoral content or more. So two results, high agreement between SeqOne and Myriad, and a smaller number of non-conclusive results using SeqOne HRD. Then there is a result which concerns the survival result. So overall, we obtain exactly the same result, whether we use SeqOne HRD or Myriad My Choice to determine the HRD subgroup. So for the HRD negative subgroup, there is no difference between the olaparib arm and the placebo arm, and it's the same negative, the same absence of difference between arm, whether we consider the Myriad or SeqOne to form the HRD subgroup. Then for the HRD positive subgroup, there is, as mentioned by Professor Pujol, clearly an advantage in terms of PFS when being in the olaparib arm. And this advantage is the same whether we consider SeqOne HRD or Myriad My Choice to form the HRD subgroup. That's an important result. The survival result are unchanged whether we consider Myriad or SeqOne to form the HRD subgroup. And then we zoom on the BRCA wild-type patient, for which HRD biomarker is very relevant. And again, we have exactly the same survival result, whether we use SeqOne HRD or Myriad My Choice. So for the negative patient, there is no effect, no advantage of being in the olaparib arm. That's the reason why the hazard ratio is equal to 1. Whereas when being in the olaparib arm is advantageous when the patient is HRD positive, with hazard ratio between 0.45 and 0.50. And the same advantage whether we consider a Myriad or SeqOne to form the HRD subgroup. So same result as before, but this time for the BRCA wild-type patient. So it's what we provide as in SeqOne is a technology agnostic approach. We have performed validation experiment, and the results are unchanged. Whether we consider Illumina NextSeq or Illumina NovaSeq to do the sequencing, we have also the same results whether we use like different type of DNA region kits provided by Agilent, XTHS1 and XTHS2. Same result with two different workflows to prepare the NGS, to perform the NGS pipeline. And right now, the clinical validation has been performed with the Agilent panel to determine BRCA1 and BRCA2 variants, but it can be done with another panel for lab which have custom panel, for example. We need a step which is a technical validation with 50 samples to validate another panel. And in terms of requirement, as I said before, we can handle 20% of cellularity, at least 50 nanogram of DNA, a minimum of 0.1x for coverage, and 1x is preferable and more optimal. And we have also a quality check to check that the quality of sequencing is good enough. In terms of quantitative landscape, competitive landscape, sorry. So here it's 3 solutions, SeqOne HRD, Myriad, and SOPHIA, which all of them have been validated with the Paola-1 reference cohort. And as I said before, we can handle a cellularity of 20% or more, whereas for the other solution, they need at least cellularity of 30% for Myriad and 28.5% for SOPHIA. And this, the fact that we can handle low cellularity explain why we have a small non-conclusive rate, 1% for SeqOne, whereas it's 4 or 7% for the other solution. And all of these solutions, Myriad, SOPHIA, and SeqOne, they all now have a decentralized solution, which is convenient for biology and pathology lab. So in conclusion, we have shown that with SeqOne, we have the same survival result compared to the standard test. We have less non-conclusive compared to the standard test because we can handle smaller values of cellularity. And we have validated, clinically validated SeqOne HRD using the Paola-1 reference cohort. It's a solution which is easy to implement for pathology and biology lab. It doesn't disrupt the current manner to do the BRCA1 and BRCA2 testing. And because there is no disruption, it's a solution which can be implemented with a low upfront financial and human cost. And it's technology agnostic. It can be used with different sequencer and different wet lab. So I thank you for your attention, and I leave the stage to Ben, who is going to moderate the Q&A session. Thanks a lot for your attention.
Thank you, Pascal and Michael, for those excellent presentations. We'll now begin the Q&A portion of the webinar. A reminder to attendees, you may type in a question at any time through the Q&A panel, which appears on the right side of the webinar presentation. One moment while we gather questions. Our first question today for Pascal and Michael. What is your threshold for CCNE1 AMP calls?
Okay, thanks for the question. I will take care of this question. So the question is, do we use a hard threshold for CCNE1? So we don't use a hard threshold because it's a predictive model in which we use a continuous value of the number of amplification at CCNE1. So there is no hard threshold for CCNE1.
Thank you. Our next question. Did your validation occur on actual samples from the PAOLA-1 trial?
Yeah, that's correct. The validation comes from 368 samples from the PAOLA-1 trial. Yeah.
Thank you. Our next question. Are structural variants, not CNA, possible to detect using the shallow whole genome sequencing platform?
So the question is about using structural variation to detect HRD. So here, what is done in the solution is to count the number of large-scale rearrangement, but using the shallow whole genome sequencing. And in the panel, we use like short indel, not large-scale rearrangement, short indels and SNV to detect BRCA1 and BRCA2 genomic alteration.
Thank you. One moment while I gather questions. I'd like to remind attendees that you may type in a question at any time through the Q&A panel on the right side of the webinar presentation. Our next question. Do you measure BRCA1 and 2 deletion as well as mutation?
Yeah, so as I just said, we use the gene panel to call genomic alteration in BRCA1 and BRCA2, and we detect a single nucleotide variant plus short insertion deletion, short indel. So yes, short deletion are detected when we use a gene panel to call the alteration.
Thank you. Our next question. How do you ensure that samples analyzed by your solution reach the minimum tumor content required? In other words, 20%, this person says.
So we have validated the 20% threshold for cellularity. It's possible that the method would be usable with a lower threshold, but right now we have validated using biological experiment the 20% threshold. So that's the threshold which has been validated with the biological experiment.
Thank you. One moment while we gather questions. Our next question. Based on your experience, what percentage of samples are BRCA positive, and therefore there is no need to run the whole genome sequencing?
Like I would say around 15% of the sample would be BRCA positive. So for this sample, there is no need of running the shallow whole genome sequencing.
Usually in the literature, I mean, it's around 15 to 20% at germline level plus 5 to 7% at somatic level.
And also, I just want something which is a bit operational, and like for some reason, it might be effective sometimes to do the gene panel and the shallow whole genome at the same time in order to save time. So there are like two options for operational aspect: either to do it in a sequential approach, starting with the BRCA testing, or in a parallel approach to save.
That's an important question, Michael, and. Can I ask you, actually, this is a question because, I mean, sequential will be the best in terms of cost. But, I mean, there is a turnaround question. How long is your turnaround time?
So typically, if you do it in parallel, if you start DNA extraction at the beginning of the week, business week, you get the HRD result at the end of the business week. So if you do it in parallel. And if you do it in a sequential approach, you have to wait till the midweek, the week after you start the DNA extraction. That's a typical turnaround time. Yeah.
That's right.
Thank you. One moment while we gather questions. Our next question, do you ever use RNA data to call HRD? And if so, how do you use it?
At least for the SeqOne test, I'm not going to answer in general, but for the sequential, for the SeqOne test, it's based on DNA only, no RNA at all. But there might be other tests doing that, but not the one I described.
Thank you. Our next question, are you able to detect large rearrangements of BRCA genes from formalin-fixed paraffin-embedded tissue?
So I think there was another question also related to this large rearrangement. So again, we didn't look for a large rearrangement when we called the genomic alteration in BRCA1 and BRCA2. We restrict the detection of genomic alteration to small to medium-sized indents.
Thank you. Our next question. When might you expect an HRD test to be approved for breast, pancreatic, and prostate cancers?
That's a good question. Yeah, thank you, Michael. I mean, I think that's an under-investigated point. I mean, we need to go beyond BRCA mutation in pancreas and prostate cancer, even if it doesn't look to be the same. We need to address this question. And I'm a little bit disappointed that there is no, at present time, trials on HRD in prostate cancer. We have a lot of studies coming out, actually, in HRR genes panel in prostate cancer, but unfortunately not in using HRD. And I think that will be a very important endpoint to address in the next future.
Thank you. Our next question, how many samples do you sequence for per patient? Just one per patient?
I'm not sure I understand the question. So you need a tumor sample, so one tumor sample, and based on this tumor sample, you have to do at the same time the gene, or at the same time, or in the same flow cell or in two different flow cells, to do the gene panel and the shallow whole genome. But it's based on the same specimen and the same tumor sample.
Thank Thank you. Our next question. In BRCA germline mutant patients, can you call tumor BRCA LOH status?
Tumor BRCA LOH status. So we don't compute LOH per se. So we have another measure, which is a loss of parental copy, which is a bit different from the LOH. We don't compute LOH.
That's an interesting point. I think behind that, there is the idea of double hit, actually, events. And it might be that there is some tumor in which it could be important to verify a double hit. And I think that's a very tricky question.
Thank you. Our next question. How is it possible to have such a low rate of NC results?
Yeah.
I'm not sure what NC.
Non-conclusive, Ben. It's a rate of non-conclusive. Yeah, the reason for that is that we have trained the model on samples which have been computationally diluted, so that the model know how to handle samples with low cellularity, low percentage of tumoral compound. So we have trained the model with low cellularity samples, and then we have done, as I mentioned before, the biological validation on this on 20% of sample with 20% of cellularity. So clearly, the low cellularity explains the fact that we can handle low cellularity samples. Explain when we have this small rate of non-conclusive.
I will add, Ben, that it is a very important question. I mean, non-contributive, non-conclusive HRD patient could be a loss of chances.
Thank you. One moment while we gather questions. Our next question, how many chemistries or sequencers have you used to process the Paolo-1 samples in the context of IVD validation?
So we have validated the test with two DNA region kits from Agilent, XTHS1 and XTHS2. And we have also validated the test using two Illumina sequencer, NextSeq and NovaSeq. So two DNA kits and two sequencers. And if a lab wants to use, for instance, another kit, we can perform a technical validation for purpose.
Thank you. Our next question, is there a minimum number of samples that can be processed simultaneously?
That's a very technical question, which depends on how you set up the process in the lab. So typically, we have a situation, if you have, let's say, a mid-output flow cell, you can typically process, the minimum would be like to have like, you can process 5 patients a week, in which you do at the same time the gene panel and the shallow whole genome sequencing in the same flow cell. And if you do that, you can process 5 patients a week within the same flow cell. And if you have less, and if you want to use a mid-output flow cell, you have to wait till you have enough patient or enough sample to process.
Thank you. Our next question. What is the recommended coverage to conduct shallow WGS sequencing, whole genome sequencing?
1x coverage. That's what we recommend for optimal results. Yeah.
Thank you. One moment while we gather questions. Can the HRD score change with time in the same patient?
Pascal, do you want to handle this question?
This is a very difficult question, actually, but it looks like HRD is a bit constant in tumor progression. As far as we could understand it, it looks like we are talking about genomic score. And so once the defect is there, I mean, it's forever, I would say. One question is, could it happen in metastatic recurrences? But I think even if it's happened because of the phylogenetic evolution, unfortunately, it's a marginal clinical point. And I would say, Barley, that the answer is no, as far as we have data on that.
Thank you. Our next question. This is in relation to a previous question. Can you elaborate on the difference between loss of heterozygosity and loss of parental copy?
Yeah, sure. So loss of heterozygosity is more general than the loss of parental copy. You can lose heterozygosity for various reasons and various situations. And whereas the loss of parental copy is only one of these reasons why you can lose. That's a subset of all the possible reasons why you can lose heterozygosity.
Thank you. Our next question. Is there a reason for not including other HRR genes in your gene panel?
I think Pascal provides some answer to that during his talk, because basically it has been found that mostly beside BRCA1 and BRCA2, like they are poorly informative about the response to PARP inhibitor, with the exception of a few genes, as shown by the Professor Pujol. But these few other genes, usually, like the mutation for these few other genes, is really weak. So that explains why we stick to BRCA1 and BRCA2 genomic alteration.
I can add something, Ben. I totally agree, and just go back to the paper by Eric Pujol, but we know already that for a lot of studies. Actually, it was not very modest to think that our understanding of HRD could allow us to see it, looking at HR genes. I mean, we just have to be modest about that. We just know a little about all the process of repair, including HR. So for me, it's not very surprising that we have a lot of missing information if we are looking at HR. But something very important for clinicians is that we cannot use HR gene panel to define HRD so far. Think about that outside BRCA1 and BRCA2 gene, we only got 2 to 3% of the 20 missing HRD patients, 2 to 3%, and mostly located on RAD51C, PALB2, and BRIP1 genes. So again, HR gene panel is not sufficient to look at HRD.
Thank you. Our next question. How many samples were used to train your model?
Around 150 in different centers.
Thank you. Our next question. Is the raw Paula-1 data publicly available?
It's not publicly available. There is an initiative led by Eric Pujol-Lorraine to evaluate both academic tests and commercial tests such as the one which have been presented. But it's not publicly available.
Thank you. Our next question. Do you confirm loss of heterozygosity for BRCA1 and BRCA2 mutated patients? This attendee says otherwise the patients are unlikely to respond. There's a second part to the question, but perhaps you can address that first.
So can you repeat, Ben, please?
Yes. Do you confirm loss of heterozygosity for BRCA1 and BRCA2 mutated patients? Otherwise, they are unlikely to respond.
So it really depends. So first of all, we don't compute loss of heterozygosity per se, but I understand the question. The question is, do we do the genomic instability for the BRCA mutated patient? It depends on the lab workflow. For some lab workflows, if the patient is BRCA mutated, it's enough, and then the patient will be eligible for the PARP inhibitor, and it corresponds, for example, to the current EMA guideline for treatment.
Michael, I think the question is about the double hit event on the BRCA1 or BRCA2 copies. You know, I think the question is very good one. Again, is there any possibility that some tumor lost one allele and not the other, so they could be less sensitive to PARP inhibitor? I think that's an open question. I think
that's a research question. I think. Yes. That's a research question, whereas here what we do provide is a test for a clinical application.
Absolutely, because we have to remind that we have non-responder to PARP inhibitor, even in HRD or in exclusive tumor mutation. So it may be some clue, and we still have to look at this question. I think it's a very good one.
Thank you. A follow-up to that question from the same attendee is, would you consider including more DDR gene variants in your assay for more genes than BRCA1 and BRCA2?
So I think it's related to the previous question about the fact that why didn't we test other HRR genes? And so again, as shown by Professor Pujol, clearly most of the other genes in HRR are bad at predicting the HRD status. And again, it has been shown in this recent paper by Eric Pujol-Loren that they are not, most of the HRR genes, besides BRCA1 and BRCA2, are poor predictor of PARP inhibitor response.
I would say, Michael, to add, for research purpose, I mean, we should keep an eye on other genes to predict HRD. But if we want to look precisely so far to HRD, we have to use one of the tools looking at genomic instability or other way to look at HRD. HR gene panel are not sufficient to do that, clearly.
Thank you. Our next question, what recommendations towards follow-up germline testing do you include in your report for samples where a BRCA mutation has been identified?
That's more, that's very country dependent, the guideline of do you do germline testing once the tumor testing has been done? So that's something which is really, in our report, if we have found a BRCA genomic alteration, it's going to be reported in the report. But then depending on the clinical guideline, there might be germline testing which might be recommended depending on the country-specific guidelines.
Thank you. That is all the time we have today for questions. If we didn't have time to get to your question, we will try to follow up with our expert panelists. We'd like to thank our speakers, Pascal Pujol of University Hospital of Montpellier and Michael Blum of SeqOne Genomics, as well as our sponsor, SeqOne Genomics. As a reminder, please look out for the survey after you log out to provide your feedback. If you missed any part of this webinar or would like to listen to it again, an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.